अस्सलाम वालेकुम स्टूडेंट्स, होपिंग यू विल बी फाइन। तो जनाब लास्ट लेक्चर हमारा था अपॉन डाइसैक्राइड्स। व्हाट आर द डाइसैक्राइड्स? शाबाश, वेरी गुड, वेरी गुड। In which two subunits are attached, in which upon the hydrolysis we get the two subunits of the monosaccharides. What are the oligosaccharides? कोशिश करें। Okay, good. In which we can get up to the 10 or up to the 20, mostly less than 10 subunits from the hydrolysis of the molecules. What is the alpha and beta linkage? शाबाश, कौन बताएगा? What is the alpha and the beta linkage? In which the alpha animal is involved, make the alpha linkage, and which the beta animal is involved, it is the beta linkage. और कितनी टाइप्स की ग्लाइकोसिडेट लिंकेजेस होती हैं? कोशिश करें, शाबाश, याद करें। दो तरह की ग्लाइकोसिडिक लिंकेजेस होती हैं। नंबर वन इज द अल्फा ग्लाइकोसिडिक लिंकेज एंड द सेकंड वन इज द one is the O glycosidic linkage and second one is the N glycosidic linkage. Very good, very good. Keep it up. Today we will discuss about the polysaccharides. Our next topic will be the polysaccharide. Serving as a monomeric unit, the monosaccharides and their derivatives are linked together to form a wide variety of the polysaccharides. They make a wide variety of the polysaccharides. The stability of the variety of the glycosidic bonds make it possible for monosaccharides to combine into the structurally distinct and the biologically useful polymers. To define the structure of the polysaccharide, several structural features must be recognized. Number one, the identity of the monomeric subunit. Number two, the sequence of the monosaccharide subunit in which they are attached. The types of the glycosidic bonds they are connected with each other. The approximate length of the chain. What is the length of the chain of the molecule? The degree of the branching. How much this molecule is branched. हम कुछ बेसिस पे पॉलीसेक्राइड्स को डिवाइड करते हैं। दो बेसिस मेजरली हम यूज करते हैं, एक फंक्शन और एक यूनिट। फंक्शन क्या है उसका और किस तरह के यूनिट्स अटैच्ड हैं। ऑन द बेसिस ऑफ यूनिट दो टाइप्स हैं पॉलीसेक्राइड की, या तो वो होमोपॉलीमर होंगे या फिर वो हेटेरोपॉलीसेक्राइड्स होंगे। होमोपॉलीसेक्राइड्स होंगे या फिर हेटेरोपॉलीसेक्राइड्स होंगे। Homopolysaccharides are composed of a single type of the monosaccharides subunit. Homopolysaccharides में single type of monosaccharide subunits attached होती हैं। Heteropolysaccharides में more than two types, ये two types की जो monosaccharide subunits हैं वो attached होती हैं। Okay, moving ahead. As we all already know that the term oligosaccharide is used to denote the polysaccharide with a small number of monosaccharides, usually fewer than the 10 subunits. Glucose and its derivatives are the most common monomeric subunits. However, other monosaccharides include fructose, galactose, and derivatives. Galactose and the derivatives are found in natural polysaccharides. In some polysaccharides, the individual strands are cross-linked by short peptides, like the peptidoglycans. हम पढ़ेंगे इसके बारे में. Storage polysaccharides. Plants and animals store energy molecule glucose like the glucose in starch and the glycogen respectively. Plants में starch होता है, animals में glycogen बनता है. These polysaccharides are stored in cell in the cytoplasmic packages called the granules. Here you can see the plant cell is the plant cell and the B is the animal cell. In the plant cells, the green dots, the green granules are storing the starch, and in the animals, the pink granules are storing the granules. This is usually in the this diagram is the actually is the diagram from the liver cells. Now moving toward the starch. A mixture of the two types of the polymeric glucose subunits. A mixture of the two types of the polymeric glucose. दो तरह की चेन्स बनाता है जिस स्टार्च में ग्लूकोज. One is the amylose and second one is the amylopectin. When starch is ingested in the human diet, its degradation begins in the mouth. The slippery enzyme alpha amylase catalyzes the hydrolysis of the glycosidic bonds, yielding the disaccharide maltose and the oligosaccharide product. These are the diagrams of the amylose and the amylopectin. Amylopectin are mostly the linear molecule. Amylose is mostly the linear molecule. It gives blue color when reacted with the iodine, because the iodine occupies the position in the helical coil of the glucose subunits. Amylose is composed on the D-glucose subunit linear in a linear fashion. Mostly it contains the alpha-1 fold linkage. Reducing and the non-reducing ends are present. Molecular weight of this molecule is fewer than is almost around 150,000 subunits, 150,000. It can exceed to some extent up to the 500,000, but mostly it is in this range. The second molecule is the amylopectin, which gives the purplish or the reddish color when treated with the iodine, which is mostly branched molecule. And this is actually a branched molecule. It is highly branched. It contains the short chains. It contains the short chains. Up to the after each 25th to 30th subunit, a branch is emerged, a branch is evolved. It contains alpha-1,4 and alpha-1,6 glycosidic linkage. It contains the alpha-1,6 glycosidic linkage. It contains both the alpha-1,4 and the alpha-1,6 linkage. Alpha-1,4 linkage in the amylopectin is present where linear chain is present, and where the branch is being emerged, over there alpha-1,6 linkage is present. It is also a molecule contains more than 500,000 molecular weight. This is the diagram showing the. This diagram is actually showing the amylose and amylopectin molecules. This is structure of the, say for example, the amylose. Here you can see alpha-1,4 linkage is present. This is the structure of the amylopectin and the glycogen. Here you can see after each 25th glucose residue, after each 25th or 25th to 30 glucose residue, a branch is present. Here the branch is present. In this branch, instead of alpha-1,4 linkage, this branch contains alpha-1,6 glycosidic linkage. Alpha-1,6 glycosidic linkage. The second molecule is glycogen. Before going towards the glycogen, a little bit about the structure. The structure of this starch can be studied by exhaustive methylation, oxidizing agent, and enzymatic action. Mostly the enzymatic action is used because it is much easier as compared to the other procedures. Alpha amylase is present in the animal digestive tract. It hydrolyzes the linear alpha-1,4 linkage of the molecule amylose. Mostly, this is responsible to attack randomly. It attacks randomly anywhere in the chain and produce the glucose and the maltose. Beta amylase, it is present in the plant, attack non-reducing end of the amylose and yield the maltose. The third one is the debranching enzyme, which cleave the alpha-1,6 linkage and hydrolyze the bonds at the branching point. For the amylopectin to complete hydrolyze the amylopectin, we mostly use the mixture of all these alpha, beta amylases and debranching enzymes. The next molecule is the glycogen. Animals store the glycogen for energy metabolism in the highly branched polymer of the highly branched polymer called the glycogen. This is the structure of the glycogen is almost resembled with the amylopectin. Both are almost similar. Both are almost similar because they are both of these contain alpha-1,4 and alpha-1,6. But difference is what is the difference? The difference is the glycogen is more branched as compared to the amylopectin. After each 8 to 10 subunit, a branch is emerged. This is the alpha-1,6 linkage, and this is the alpha-1,4 linkage. Alpha-1,4 linkage makes the linear molecule. Alpha-1,6 linkage make the branch on the molecule. They are responsible to make the branch. Like the amylopectin, Glycogen consists of single reducing end and many non-reducing end. Why these reducing and non-reducing end are required? This is of the significance in the mobilization of the glucose unit for the energy metabolism. The enzyme glycogen phosphorylase is responsible to catalyze the removal of glucose residue from the numerous non-reducing ends, providing abundant supplies of the glucose when necessary. When the metabolism of the glycogen is necessary over there, this uh, reducing and non-reducing are ends become very important, because I already told you that alpha amylase, that beta amylase attack on the non-reducing end. Similarly, this enzyme called glycogen phosphorylase also recognize non-reducing end to yield the glucose subunit. Although glycogen is present both in liver and the muscle cells, it is much more abundant in the hepatic cells, where it may account for as much as 10% of the weight and only about 1% of the weight of the muscle cells in the glycogen. The extended chain and the angle of the alpha 1,4 glycosidic bond of the amylose amylopectin in the glycogen make it possible for them to fold into the tightly coiled helical structure. The structure of the molecule. This is the tight helical structure of these molecules. Approximately six in this helical, approximately six glucose residue make up 1 ton of the helix. Other minor form of the storage polysaccharides are present in nature, like the dextrin found in the yeast and the bacteria consist of the glucose residue connecting into a main chain via alpha 1,6 linkage, and the glycosidic bonds with the occasional branches formed by alpha 1,2, 1,3 and the 1,4 glycosidic bonds are also present. Bacteria growing on the teeth produce extracellular dextrin that accumulate and become an important component of the dental plaque. Inulin, a homopolymer of the D-fructose connected by the beta 1,2 glycosidic linkages. It is found in the artichokes and the other vegetables. Now, the second form of the polysaccharides are the structural polysaccharides. Some polysaccharides, such as cellulose, chitin, mucopolysaccharides, are synthesized inside the cells but extruded to the outside to provide a protective wall or lubricative coating to cells. First, हम discuss करेंगे about the cellulose. This is the major structural component of the wood and the plant fiber. This glucose polymer is extremely abundant in nature. It makes around 50% of the organic matter of biosphere. It is unbranched polymer connecting D-glucose units together. The glycosidic linkage which is present in this molecule is mostly beta 1,4. This is the structure of the, say for example, cellulose molecule. Uh, Other than the beta 1,4 linkage, there is also a cross-linking hydrogen bonds are also present in this molecule. These hydrogen bonding can be intra and intermolecular. Because of this hydrogen bonding, it makes a strong and rigid network, and this rigid network provides the scaffolding for the plant cell wall. This strong and stabilized sheet-like arrangement is useful and make this product better for the cloth industry, for the paper making, for cardboards and the building material. Although a portion of our diet consists of the vegetables and the fruit containing the cellulose, we are unable to extract the cellular energy from this glucose polymer. Animals do not produce the enzyme that can catalyze the hydrolysis of the beta 1,4 glycosidic bond between the glucose residues in the cellulose. Some organisms contain the ability to digest the beta 1,4 linkage because they contain the enzyme cellulase. Like the wood rotten fungi and some bacteria obtain nutrient glucose by synthesizing and secreting enzymes that catalyze the hydrolysis of the beta 1,4. These organisms are largely responsible for decaying of dead wood in the forest. Ruminant animals like the cattle, sheep, goat, camels, and the giraffe, these are also able to use cellulose Why they can use the cellulose? Because as a nutrient source, because they're uh, how these animals can use the cellulose, because their second stomach contain the bacteria that secrete the cellulose. In similar fashion, termites can digest the wood because they can also contain the because their intestinal tract contain the trichomypha, a symbiotic microorganism that produce the cellulose. Cellulose and its derivatives do, however, perform an essential role in the diet of the animal as bulk fiber uh, or roughage to assist the digestion and absorption of the nutrients. Another important polysaccharide component of the plant cell wall is the pectin, a polyuronic acid, a C4 epimer of the glucose derivative, D-glucuronic acid. Now we are going to study the pectin. This is extracted from the plant as used as the gelling agent in the preparation of the jams and the jellies. This is the structure of the glucuronic acid. The protective exoskeleton of the arthropods, insects, crabs, etc. And this is made up of the chitin. This animal is the crab. It contains the chitin. This polymer is also found in smaller amount in the cell wall of the yeast, fungi, and the algae. The monomeric building block of the chitin is the glucose derivative, N-acetyl glucosamine, linked in a beta 1,4 glycosidic bonds. These are the two molecules. 1,4 N-acetyl glucosamines are linked in the chitin in beta 1,4 linkage. Like cellulose, chitin exists in extended chain that are associated into the fiber by intra and intermolecular hydrogen bonds. Because human has no enzyme that can catalyze the hydrolysis of the beta 1,4 linkage in the chitin, it is indigestible. Because we have no enzyme for the beta 1,4 digestion, so we can't digest this molecule. Now we are moving toward the structural polysaccharides. These are also found in the connective tissues, cartilage and tendons, or extracellular matrices. Ground substances of the higher animals. The extracellular matrix is a gel-like material that act as glue to hold the cells together. Structural polysaccharides are present in the bones and the cartilages and in the cell connecting, and it also make the cell connective connecting material. A group of polymers called the mucopolysaccharides provide a thin viscous jelly-like coating to the cells. The most abundant in this class of the polysaccharide is the hyaluronic acid, which has alternating monomeric unit of the N-acetyl glucosamine and the D-glucuronic acid. The structure of this molecule is very interesting. Very interesting. I told you that there are the two molecules, two types of the two types of the monosaccharides are present. One is the glucuronic acid. So, for example, this one is the glucuronic acid, and this one is the N-acetyl glucosamine. The glucuronic acid is attached with the N-acetyl glucosamine through beta 1,3 linkage. Here you can see the beta 1,3 linkage. And after attaching with this molecule, it makes a disaccharide. It makes a disaccharide. Similarly, there is another unit of glucose and D-glucuronic acid and the N-acetyl glucosamine. This is another, say for example, disaccharide molecule. Okay. Now there are two disaccharide molecules attached with one another through beta, in which two monosaccharide subunits are attached to beta 1,3. Now this disaccharide is attached to this disaccharide through beta 1,4 linkage, to beta 1,4 linkage. So we can say that glucuronic acid is attached with the N-acetyl glucosamine to the 1,3 linkage, and N-acetylamine in turn attached to the glucuronic acid to beta 1,4. Hyaluronic acid serves as a lubricant in the synovial fluid of the joints, and is found in the extracellular matrix of the connective tissues. Another component of the extracellular matrix is the chondroitin sulfate, a heteropolymer mucopolysaccharides, the hydropolymer mucopolysaccharide. Like hyaluronic acid, it is composed of the two alternating monomeric subunit N-acetylglucose and amine sulfate and the D-glucuronic acid. It contains the C4 and C6 at the C4 and C6 position of the amino sugar. It contains the sulfate ester. As I already told you that this is important in the bones, cartilage, and the tendon. The next molecule is the structural polypeptide. It's a structural peptidoglycan. This molecule is present in the bacteria. In this molecule, two types of the molecule in this molecule. This molecule make the rigid cell wall of the bacteria, which provide the physical protection and composed primarily of an unbranched heteropolymer of the alternating N-acetylglucosamine and N-acetyluronic acid. Two molecules, the heteromolecules are polymer. This is the heteropolymer. The monomeric subunits are linked into an extended backbone. This is very important to know. Look on this structure. मैंने आपसे ये कहा है कि N-acetyl glucose अमीन और म्यूरेमिक एसिड के ये सबयूनिट बने हुए होते हैं. हर म्यूरेमिक एसिड से एक टेट्रा पेप्टाइड मर्ज होती है. हर म्यूरेमिक एसिड से एक टेट्रा पेप्टाइड जो है वो निकलती है, उसके साथ अटैच होती है. ठीक है. तो there are different chains होती हैं. For example, this is chain number 1 and this is chain number 2. ये chain number 2 है. ये पैरेलल आपस में ये पैरेलल चलती आ रही है. ये पैरेलल चल रही है इसके साथ. ओके. अब यहाँ पे म्यूरेमिक एसिड से टेट्रापेप्टाइड निकल रही है और ये दूसरी इसकी लोअर चेन है, इससे एक टेट्रापेप्टाइड यहाँ से रिमूव हो रही है. ये दोनों टेट्रापेप्टाइड आपस में क्रॉस लिंक करती हैं through 5 ग्लाइसिन रेसिड्यूज. ठीक है. इन टर्न ये टेट्रापेप्टाइड एक पेंटाग्लाइसिन के थ्रू एक दूसरे के साथ अटैच होती हैं. सो द मोनोमेरिक, ये यू कैन सी अगेन आई एम टेलिंग यू, इस पर से ये टेट्रापेप्टाइड निकलती है, इस पर से ये टेट्रापेप्टाइड निकलती है. ठीक है. अब ये दोनों टेट्रापेप्टाइड एक दूसरे के साथ 5 ग्लाइसिन रेसिड्यूज के साथ अटैच होती हैं. ये ग्लाइसिन रेसिड्यूज इस तरीके से अटैच होती हैं कि एक के लाइसिन रेसिड्यू को दूसरी चेन के जो लास्ट वाला टर्मिनल एलानिन रेसिड्यू होता है उसके साथ अटैच करवा रही होती है. ओके. The monomer subunits are linked into an extended backbone by the beta-1,4 glycosidic bond. The rigidity and the strength of the bacterial cell walls are consequences of the network of the peptide cross links between the strands of linear polysaccharides. Okay. The amino acid composition and the sequence of the peptide links vary from bacterium to bacterium. Gram-positive bacterium, say for example Staphylococcus aureus, 2 sets of the peptides, a tetrapeptide and a pentapeptides of the 5 glycine residues form a cross link. So for example, this one here from this chain, so for example, the uronic acid, the 4 pentaglycines will be emerged, and this contains the 5 glycine residue, which in turn will be attached to the lysine residue of the other chain. So, structural properties of the common polysaccharides. Number 1, हमने पढ़ा आज स्टार्च को, जिसमें एमाइलोज होमोपोलीमेरिक है, एमाइलोपेक्टिन होमोपोलीमेरिक है. इसमें अल्फा 1,4 लिंकेज होती है. एमाइलोपेक्टिन में अल्फा 1,4 और 1,6 लिंकेज दोनों होती हैं. ये दोनों न्यूट्रिएंट वैल्यूज हैं और दोनों प्लांट में प्रेजेंट होते हैं. ग्लाइकोजन होमोपोलीमेरिक है. ग्लूकोज 1,4 और 1,6 लिंकेजेस में प्रेजेंट होती है और न्यूट्रिएंट स्टोरेज है और ये एनिमल में प्रेजेंट होता है. डेक्स्ट्रिन होमोपोलीमेरिक है. ग्लूकोज 1,6, 1,2, 1,3, 1,4 ब्रांचेस इसमें होती हैं. न्यूट्रिएंट स्टोरेज है, यीस्ट और बैक्टीरिया में प्रेजेंट होते हैं. इनुलिन होमोपोलीमेरिक है. फ्रुक्टोज 2,1 इसमें बीटा फ्रुक्टोज 2,1 लिंकेज इसमें प्रेजेंट होती है. न्यूट्रिएंट स्टोरेज है, प्लांट्स में प्रेजेंट होते हैं. सेलुलोज होमोपोलीमेरिक है. ग्लूकोज बीटा 1,4 लिंकेज इसमें होती है. स्ट्रक्चरल फंक्शन इन प्लांट. Pectin homopolymeric. Pectin is the homopolymeric galacturonic acid होते हैं. Structurally rigidity in the plants they provide. Chitin, these are the homopolymeric N-acetylglucosamine. These are the N-acetylglucosamine beta 1,4 linkages present in these. molecule structural function in the exoskeletons of some animals. Hyaluronic acid is the heteropolymer N-acetylglucosamine and glucuronic acids, beta 1,4 and beta 1,3 linkage is present, lubricant in the synovial fluid and extracellular matrix. Chondroitin sulfate heteropolymer N-acetylglucosamine sulfur and glucuronic acid, beta 1,3 and beta 1,4 linkage. It also provides lubrication in the synovial fluid and an extracellular matrix. Peptidoglycan heteropolymeric with the peptide crosslinks N-acetylglucosamine and N-acetylaminic acid beta 1,4 linkage होती है. They provide the structural function in the bacterial cell walls. So I hope it will be enough for today. It will be enough for all of you for today. For further, we will move tomorrow, inshallah. So best of luck for all of you. Keep enjoying your vacations and keep studying during this Corona season. Thanks a lot. Allah Hafiz.